Good morning, students. In today's session, we will be discussing about the reproduction in Cycas. So, Cycas, it undergoes a reproduction by way of vegetative and also sexual methods. Firstly, we'll see through the vegetative mode of reproduction. In vegetative reproduction, it occurs by means of adventitious buds. It is also known as bulbils. So these adventitious buds or bulbils develops from the basal part of the stem. Actually, they have the persistent leaf bases along their stem. So it develops through the crevices of these type of leaf bases. And also from the roots, suckers may also develop and they grow horizontally. They will be growing horizontal to the ground surface, and after detachment, both these adventitious buds or the bulbils and also the suckers, they after detachment from the main plant, they develop to a new plant. So this is the mode of vegetative reproduction occurring in case of Cycas. And now moving on to the sexual reproduction. Cycas, as the case is, it is a dioecious plant. So that is the male and the female organs, they are seen in separate plants. There will be a male plant and there will be a female plant. So it is dioecious. So the male reproductive organs and the female reproductive organs are there. They are the cones. The reproductive organs are known as cones or strobili. So there will be a male cone and a female cone. We'll discuss about the details of this cone subject later. So for now, there will be a male and female cone. That is, to deal with this subject, the male cones will be there as an organized structure, while in case of female plants, an organized cone will be not there. So in case of female plants, they will be having the megasporophylls, and these megasporophylls are not organized to form a cone-like structure. So first is the male cone. Male cones, they develop singly at the apex of the plant, and they will be seen in between the leaves, the foliage leaves. They will be seen in between the foliage leaves and they form a crown around. The foliage leaves, they form a crown around. So during this process, the stem will not be growing. At the time of cone development, the growth will be arrested. So that is the case, and these male cones, they are extremely small in size. That is, they will be having a stalk, and the shape will be like an oval or a conical shaped structure, and it will be 20 to 80 centimeters long. That is, it is the largest cone in the plant kingdom. So it is the largest cone in the plant kingdom, and it will be ranging from 20 to 80 centimeters in length. And many microsporophylls will be there, and they are spirally arranged around the axis. They will be spirally arranged along the cone axis in an acropetal succession. The microsporophylls are soft and fleshy, when they are young, but at the time of maturity they will be so hard, woody like structure, and they will be larger in the middle region and smaller at the base and the tip of the cone axis. So regarding each microsporophyll, a mature microsporophyll will be a somewhat triangular structure. It will be somewhat triangular structure, and they will be differentiated into a proximal wedge shaped structure, the fertile part and a distal sterile part. The distal sterile part, it tapers, and this tapering end is called as apophysis. The abaxial surface of the microsporophyll is sterile, and on its lower adaxial surface there will be numerous microsporangia arranged in groups called as sori. Each sori consists of 3 to 6 microsporangia, which will be arising from a indusial papilla. And in between the sporangia there will be numerous delicate hairs which protect this young sporangia and also help during the dehiscence of this microspores. The wall of this microsporangia are differentiated into 3 regions: exothecium, endothecium and tapetum. Exothecium is the outer wall and it is highly cutinized and thick walled. Endothecium is thin walled, and the tapetum is nutritive cells, composed of nutritive cells. And inside this sporangia they contain a large number of microspores. Each of these microspores or the pollen grains will be uninucleate and a haploid one. It will be having a thick spore wall, and this spore wall has 2 layers. The outer one is called exine and the inner one is the intine. So this is all about the male cone and microspore. Next we will be discussing about the female cones in our next session. So thank you.